Global food systems are under increasing strain from unsustainable farming practices, population growth, and especially drought caused by climate change. These pressures threaten food security worldwide. But what if we could leverage nature's own solutions to combat these challenges? Our team drew inspiration from the fascinating symbiosis between plants and microorganisms, such as biofilm-forming rhizobacteria. The biofilm formed brings many benefits to the plants. It attracts and retains water at the plant roots, reduces salt uptake, improves nutrient absorption, and provides a physical barrier against phytopathogens. In this way, biofilms are the all-rounder solution to a plant's worst nightmares, be it drought and high soil salinity, poor nutrient availability, or the attacks from phytopathogens. Our approach focuses on genetically modifying Pseudomonas IsoF to enhance biofilm production when it senses plant exudates. To contain our bacteria's engineered activity to the plant's rhizosphere, we incorporated a xylose sensing system. Xylose is a root exudate. When the bacteria sense xylose at the plant's root, a feedback loop activates, inducing the overexpression of diguanylate cyclase, or DGC. DGCs are enzymes that lead to the production of c-di-GMP, an intracellular signaling molecule. This molecule activates genes involved in extracellular polymer production, which in turn is crucial to biofilm formation. Another approach for increasing c-di-GMP is the knockdown of PDEs, using interference RNA. These enzymes degrade c-di-GMP. By knocking them down, we indirectly increase intracellular c-di-GMP. The combination of both mechanisms increases the amount of biofilm created at a plant's root. To ensure environmental safety and prevent the engineered bacteria from spreading beyond the target area, we incorporate a kill switch. It is composed of a toxin and an antitoxin, targeting the bacteria's DNA gyrase. Once activated by the xylose-induced feedback loop, the toxin production can't be stopped. The antitoxin production, however, is directly linked to the xylose sensing. Thus, when the bacterium leaves the rhizosphere and no longer detects xylose, antitoxin production is stopped, and the bacterium is killed by its ongoing toxin production. By leveraging synthetic biology, our project provides a sustainable solution that enhances the resilience of agricultural systems, ensuring food security for the future.
Once we had designed our molecular mechanism, it was important for us to think of methods of implementation. We talked with a farmer, with agricultural experts, and with a stakeholder from the agricultural biologicals industry. We found that our bacteria could be easily integrated into agrosystems using current farming practices. In the field, RhyzUp could either be applied with crop sprayers, used to inoculate seedlings, used as a seed coating, or be used in combination with microgranular fertilizers. All of these techniques are standard seeding methods. Our bacteria would simply have to be added to an appropriate liquid solution. Several stakeholders have highlighted the efficacy of products like ours in systems with low microbial diversity. This means that our project could be of particular benefit to depleted fields where microbial richness is already extremely low. Therefore, we tackle the problem where it is most urgent, at its roots.
The approach we chose to tackle this problem is by upregulating the c-di-GMP production in the cell. Its concentration is regulated by two enzyme families, the DGCs and the PDEs. Diguanylate cyclases, or DGC, synthesize c-di-GMP by taking two GTP molecules and cyclasing it into one c-di-GMP molecule. By introducing a DGC into our bacterium, we can try to increase the c-di-GMP production in the cell and thus the biofilm formation of our bacterium. c-di-GMP binds allosterically to this enzyme, or more precise, to the I site within the GGF domain, which then decreases the enzyme's activity. So when the c-di-GMP concentration in the cell is too high, it serves as a negative allosteric inhibitor and decreases the enzyme's activity. By altering the sequence of the enzyme, we can try to inhibit this feedback loop and alter the I site so that the c-di-GMP can't bind to the enzyme anymore. Using AlphaFold and sequence alignment, we created two mutants to inhibit the I site. We exchanged an arginine residue with an alanine residue, once at position 196 and once at position 242. Additionally, we wanted to test WISP-R, a DGC native to Pseudomonas aeruginosa, and also its mutant. Its mutant is already established, and they exchanged a arginine residue at position 242 with an alanine. Now we have 5 different DGCs, that we want to test in order to find out which one of those leads to the highest increase in c-di-GMP production and biofilm formation.
The second enzyme family that we're targeting in our project is that of phosphodiestrases, or PDEs. PDEs regulate the c-di-GMP level in the cell as they degrade c-di-GMP to GMP. As our goal is to heighten the c-di-GMP level, we aim to target a specific PDE, which is native to the Pseudomonas species IsoF strain. The approach which we used to knock down this PDE is that of the CRISPR-I, or CRISPR interference system. The CRISPR-I system is a modified version of the CRISPR-Cas9 gene editing system, which is designed to inhibit gene expression. The way this is done is because of the dCas9 enzyme. This dead Cas9 enzyme doesn't cut the DNA, but simply binds to the target DNA and therefore inhibits the transcription of our target gene. The dCas9 enzyme, which is already on the genome of the strain that we were provided with, is the first major component of the CRISPR-I system. The second major component is the sgRNA, or single guide RNA. This single guide RNA is made up of a tracker, which forms a hairpin structure, which is how the dCas9 enzyme binds to our target DNA. We designed this sgRNA by first identifying a PAM motif near the promoter. This PAM motif is essential for the dCas9 inhibition. Furthermore, we ensured that the nucleotides adjacent to the PAM motif were unique, therefore ensuring that there are no off-target effects or off-target binding of the dCas9 in the genome. We introduced our sgRNA into the plasmid using PCR amplification and blunt-end ligation. This way, we successfully managed to knock down our PDE of choice.
I will now continue with the results of our project. We will focus on biofilm quantification. We conducted 3 experiments. In the first experiment, we performed a cyclic DGMP assay. In our experiments, we included 3 negative controls: Pseudomonas IsoF with an empty plasmid, bifidin plasmid, and one containing a PDE overexpression. Further, we included a positive control, Pseudomonas IsoF ChatQ, which is a strain known for overexpressing biofilm. In our construct, we included a Brown-Knox promoter to test the difference when our construct is active versus inactive. As you can see in our graph, the PDE knockdown strain visibly increases the cyclic DGMP production when induced with Brown-Knox versus the non-Brown-Knox condition. Further, our own mutations, DGC R196A and R248, showed a significant increase when induced with Brown-Knox versus the no Brown-Knox condition. In the next step, we compared our constructs with a control. However, we only got a significant increase for the mutated WISP-R construct. Lastly, we compared our mutated DGCs to the DGC wild type. Our mutated construct, the DGC 196A, showed a significant increase in the cyclic DGMP level compared to the wild type DGC. This indicates that we successfully increased the DGC's activity by tackling the enzyme's I site. For our second experiment, we conducted a biofilm staining. We again included the same controls as I previously described. The PDE knockdown showed higher levels than the negative control. However, the levels were lower than our positive control, ChatQ. Moving on to our DGC strains, here we have 3 different constructs where we see a significant increase compared to the control. The strain that contains the normal DGC, the DGC WISP-R, and the mutated DGC WISP-R. The mutated WISP-R showed the highest fluorescence, even exceeding the positive control, ChatQ. Our mutated DGC, R248, showed a modest increase in fluorescence compared to the control.
Low motility has been shown to increase biofilm formation. In order to measure this aspect of our construct, we conducted a motility assay. We plated the same controls and strains as measured in the c-di-GMP assay before, but now on a semi-solid agar plate. After 8 hours of growth, we measured the diameter of the swimming motility. As expected, our negative controls consistently had a higher motility, which is disadvantageous for biofilm formation. Meanwhile, all DGCs and the PDE knockdown had a lower motility. We wanted to measure the effect of a DGC overexpression on plants. In order to do this, we inoculated tomato plants with a DGC overexpression, JETQ, a PDE overexpression, and LB and wild-type IsoF as controls. We grew these plants under 2 conditions, wet and dry. To quantify these findings, we measured the plant's wet weight of the aerial part after 2 weeks of growth. Under wet conditions, the JETQ inoculated plants had the highest wet weight. However, when measuring the dry weight, this difference became negligible. This indicates that a DGC overexpression may not increase plant growth, but rather increase its ability to retain water. We therefore speculate that a plant inoculated with a DGC overexpressing bacteria may be better equipped in the face of drought.
To advance biology projects, especially in synthetic biology, computational models play a crucial role. In our product RhyzUp, we use simulations and computational modeling to gain deeper insights into the biological process of biofilm formation, to refine our product design, and to test out implementation in field applications. While the wet lab focused on increasing biofilm production, we in the dry lab ask ourselves, what happens to the bacteria metabolically when it's forced to produce more biofilm? How far can we push this process, and how does the environment influence the metabolism? To answer these questions, we use flux balance analysis. FBA translates the metabolic network of an organism into a computer-readable format, allowing analysis with it. As there was no genome-scale metabolic model of Pseudomonas species IsoF, we based our analysis on a related species Pseudomonas putida KT2440. We used the two exapolysaccharides, alginate and cellulose, and the biomass function in the model to assess the impact of biofilm formation on bacterial growth. We analyzed this across different environments: the M9 glucose minimal medium, the LB medium, which represents our wet lab conditions, and soil, rhizosphere, and drought, which mimic our real-world application conditions. In all conditions, we observe a clear trade-off between biomass and biofilm production. When we increase biofilm formation, the biomass decreases, as nutrients such as glucose gets diverted from biomass to biofilm formation. This indicates that biofilm production is very costly for the bacteria, and therefore wet lab's approach of making the bacteria the way that it only produces hyperrobust biofilm close to plant roots is very well founded. We discovered that both the cost and the maximum quantity of biofilm production is highly dependent on the nutritional complexity of the environment. In glucose minimal medium, the relationship is very linear. In the more nutrient-rich and complex LB medium, the yield of biomass and of biofilm production is higher, and the relationship is also not quite linear. Across soil, rhizosphere, and drought, we observe similar linear trends, indicating that our bacteria is resilient to environmental changes. Due to the low availability of nutrients such as sugars or amino acids, the yield of biomass and biofilm production is much lower than in the lab conditions. We conducted a gene assay to explore whether knocking out specific genes could enhance biofilm formation without compromising growth. With that, we identified 3 genes that increase cellulose production. And these genes could then be a future target for wet lab to further improve our product. We also evaluated whether nutrient supplementation could improve biofilm formation in field applications. While polysaccharides and amino acids directly boost biofilm formation, other nutrients such as minerals had minimal impact. Therefore, we would not recommend to use supplementation, but rather use a higher concentration of our product near the plant roots to improve colonization.
Besides our exchange with experts in agriculture and ecology, we talked to a lawyer, an ethicist, NGOs, and an expert in consumer behavior. Most of these interviews were recorded. The edited versions of these videos can be found on our wiki. In this way, we are making the knowledge we have gained available to a wider audience. In addition, our team made several other contributions to education. We organized lunch lectures, high school classes, and a lab workshop. In this way, we were able to reach people of different ages. The main focus of our human practices research, however, was sustainability. This helped evaluate how our project contributes to the UN Sustainable Development Goals. In short, our bacteria improve nutrient uptake, pathogen resistance, and drought tolerance. Ultimately, this contributes to agricultural practices that are more resilient and less polluting. In this way, our approach addresses the UN goals of zero hunger, good health and well-being, clean water and sanitation, climate action, and life on land. Participating in iGEM was a blast, and if you want to learn more about our project, check out our wiki. We can't wait for the Grand Jamboree in Paris.